Good morning to all of you. So today is our fifth lecture and we are observing on the algal pigmentation. So I think we are knowing there is having the algal diversity and there is a diversity in the pigmentation. So we are already knowing there is a red, yellow, blue, these pigments they are generally found in the marine and the freshwater conditions are there. As per the different groups of the algae, as we are knowing, there is a specific pigment composition, and this pigment composition generally which are found pigments in the plastids and which are commonly called as the chromatophores, right? The distribution pattern of the pigments shows the great taxonomic significance. The classification given by Fritsch as so they have, it is totally based on the algal pigmentation. We are observing this classification. These major algal pigments they are having the certain cells are there and they are having the characteristics feature. So as in introduction, the algal plants mostly they possess the undifferentiated thallus, that is the root, stem and the leaves, or the thallus range. It is from the microscopic to the single celled organisms are there, generally seen in the freshwater and the seawater, right? So as already we are knowing, there is the specific type of the condition is there. So this specific condition as they shows the chloroplast and the chromatoplast are there, and basically the specialized body, it is called as the pyrenoid. This pyrenoid they shows the colorless mass of the proteins surrounded by the starch plants are there. While we are observing the different pigments are there is a major role of the chlorophyll is there. So chlorophyll is a green pigment generally found in the cyanobacteria and the chloroplast of the algae. The plant algae and the plants, generally the chlorophyll they having the capacity to absorb the sunlight, and generally they are supposed to be, there is a blue portion of the observing in the electromagnetic spectrum. Electromagnetic spectrum we are knowing VIBGYOR, that is the violet, indigo, green, yellow, red, yellow, orange and the red. So red having the maximum light intensity. So there is a blue color is maximum observed by this chlorophyll. Then there is a having the, according to the one authority, Richard Willstar and the Hans Fischer, so they have shown there is having the certain type of the process is there. Then the structures based on the chlorine ring at the center of each magnesium oil, and this structure generally they show the side chains depending on the type of the chlorophyll. Already we are knowing the chlorophylls are there, so they show the pigments, the chemical compounds, and they having the different wavelengths are there. So they having of the visible light and therefore the colorful. The flowers, corals and the animal skin contain pigments and they having the different colors are there. So reflection of light is the ability of the pigments to absorb the certain wavelength. A photosynthetic pigment which acting as an accessory pigment, chloroplast pigment or antenna pigment, they are also called, and they present in the chloroplast and the photosynthetic organisms which capture the light. So through the photon, in algae commonly the pigments found are the chlorophyll, xanthophyll, phycoxanthin, phycocyanin, phycoerythrin. So we have to discuss this general character. Generally, chlorophylls are responsible for the green color of the algae and the other higher plants. So chlorophyll A, you see that these formulas are there. Chlorophyll A, C55, H72O5, N4, Mg. Same thing for chlorophyll B, C, then chlorophyll D, E, and F is there. So in different algae, their nature is different, and there are the chlorines which absorb the blue region, which reflect from the light. The distribution of chlorophyll in algal groups. So chlorophyll A commonly present in all algae. Chlorophyll B present in chlorophyta, green algae. Chlorophyll C present in bacillariophyta, that is diatoms. Chlorophyll D present in rhodophyceae, red algae. Chlorophyll E present in xanthophyceae, and chlorophyll F, it is discovered from the stromatolites, that is the near to the these members of the diatoms. This is the general structure of the chlorophyll, and their functions are there. So already we have seen the chlorophyll. Generally they are used in oxygenic photosynthetic synthesis, and this photosynthesis commonly seen in eukaryotes and the cyanobacteria, then prochlorophytes, due to their role in the primary electron donor, that is the electron transport chain is there. Chlorophyll which absorb also transfer the resonance energy in the antenna complex, and therefore this having the importance. Then within the reaction center, both generally here as per the chemical its nature structure, so they shows the transport chain through the redox reaction. So we should know this structure. Same thing for chlorophyll B, as it is to be having the green algae, and which absorb effectively at the blue 470, then 430 and 640. So these wavelengths of the lights, they are very important. Then it is accessory pigment, photosynthetic pigment, and generally it shows heterocyclic compounds. The main function is chlorophyll. It does not absorb the light, but functions as a light harvesting antenna in the photosystem. So it is the main character. Then xanthophylls are there. These are yellow pigments. Generally they are the two major groups of carotenoids. C40 terpenoid compound, which is formed by condensation of isoprene unit is there. The main function of this it is xanthophylls act as an accessory light harvesting pigment, and this shows the critical structure and the functional role in the photosynthesis. Right, so you remember this. Mainly work as an antioxidant, and xanthophylls may be involved in the inhibiting lipid peroxidation. Right, lipid, that is it is a part of the fat. Then ficoxanthin, it is a carotenoid which performs limited form, and this generally as it is olive range in the olive green color. Then molecular formula C42H58O6. It absorbs the red light which perform the photosynthesis, and abundant quantities there are we are knowing edible. Some brown algae are there, lamina ria, or the India ria, or sargassum, and these xanthophylls generally they shows the carotenoid. Right, so just we have to maintain this character. The function of xanthophyll generally, as they are the oxygenated carotene and the plant pigments mainly in this brown algae commonly seen. The wavelength of light generally differ from this one. Then phycocyanin, it is a pigment protein complex from the light harvesting phycobilioprotein, which is along with the allophycocyanin and the phycoerythrin. The water soluble, and they exist through the membrane of the carotenoids. Right. Function of phycocyanin, as it is to be there. So it shows in the different variety of algae, and generally there is it is acting as a hepatoprotective effect, and which is experimentally proved. Secondly, there is in the carbon tetrachloride is there. It reduces carbon tetrachloride to the lipid peroxidation. So in vivo experimentation, right? So these are the some characters for the pigmentation. Nextly, already all others are there. We are knowing the phycoerythrin. So it is also the same. It is a red protein pigment complex from the light harvesting of phycobilioprotein. So just you remember this character. They have also as an accessory pigment. The formulas they are not important, but for our understanding. Then pigments in the algae. So carotenoids are there. We are knowing in the carrot or the other flowers they are present. So these are fat soluble yellow pigments in association with chlorophyll. Then they protect the chlorophylls from the photo damage. Chemically carotenoids are the terpenoids. So like this one. Then vitamin A and C. Then pigments in algae we are knowing like the carotenoids, carotenes is seen in the tomato, but here these are the commonly also seen in the algal members are there. So beta carotene, lycopene are the good examples. Then pigments in algae, there are the xanthophylls are there. We are knowing the xanthophylls commonly seen in egg albumin, and xanthophylls are also called as carotenes. They are commonly seen from the algal source are there. Then phycobilins are there. They are the water soluble, and they shows there are the water soluble proteins are there. These are called as the phycobilioproteins. They are generally blue and red colored, right? So just we have to see this, and therefore nowadays in Japan maximum food is seen from the algal product algae. Then phycobilins are there, so they are in the phycocyanin and the phycoerythrin. So these are the some about the other pigments present in the algae. Then in pigments in algae, major algal groups: chlorophyll, chlorophyta, xanthophyce, bacillophyce. So chlorophyll A, B, beta carotene, these are the common, right? So just you remember all these one. In the next lecture we will say about the reserve food and the perination mode. Okay, so thanks to all.